Let us discuss the topic DNA library, also known as gene library. Now, what is this DNA library or gene library? As we all know that library is a place where we used to collect books. Same, gene library or DNA library is the collection of different DNA sequences from an organism where each sequence has been cloned into a vector. Means each sequence, if there is any DNA sequence, that sequence has been cloned into a vector for ease of purification, its storage, for ease of purification, of course storage, and next is for its analysis. If we want to analyze any particular genome or any DNA sequence, so we can analyze it easily from our DNA library. On the basis of source DNA use, this DNA library is of two types. What are these two types? As I already told you that these two types are on the basis of their source DNA. So first is your genomic library. Another one is cDNA library. Now what is this genomic and cDNA library? Genomic library, where whole genomic DNA is used, that we'll use the genomic DNA for construction of library, while cDNA library, where we use copy DNA, cDNA, or complementary DNA for producing our library, for making our library. Now what is this cDNA? Question arises that what is the cDNA? So we'll discuss cDNA afterwards, because first of all we are going to deal with genomic DNA library. So first of all we'll discuss about genomic library. That what is this genomic library? Genomic library. As I have already told that it is a collection of clones that represent the complete genome of an organism. Complete genomic. Genome means sum total of all the genes. So genomic library is the library where all the genes are present. All fragments of DNA inserted into vectors for further propagation. In genomic library, we'll insert all the fragments of DNA into vector so that they will further propagate into suitable host. For construction of genomic library, whole DNA means the entire genomic DNA is isolated from the cell or a tissue, and then it is purified and randomly break into fragments of correct size. I'll show you how can we prepare a genomic library. See, for example, if it is a cell, any cell from which we are going to isolate DNA. Clear? For example, this is a pancreatic cell. So any cell from where we are going to isolate DNA. So what we'll do, we'll isolate this DNA. This is the DNA of this cell. We'll isolate this DNA by different methods. After isolation of the DNA, by using restriction enzymes, we'll do partial restriction digestion. Means we will use restriction enzymes for the digestion of this DNA. Now, for example, after using restriction enzymes, we'll get these 6 fragments. Let us number them: 1, 2, 3, 4, 5, 6. For example, we'll get this 6 fragments. These are DNA fragments after shearing. Means this is a cell, we have isolated the DNA. After that, by using restriction enzymes, we will cut these DNA into different fragments. These fragments are generated by restriction enzyme. Now come here. For example, there is one more cell, that is E. coli cell. E. coli cell. These are the vectors, plasmids of E. coli. Already present in a cell. This is the nuclear DNA of E. coli. Now we will isolate these plasmids from E. coli. Plasmids are circular DNA. all of us know about the plasmids, which are circular DNA. We'll isolate these plasmids from E. coli cell. Like this, from different E. coli cells, not only from single E. coli cell, from many E. coli cells, isolation of plasmids. We'll done isolation of plasmids. Now use same restriction enzymes for these plasmids to cut these plasmids. So same restriction enzyme, the enzyme which we used in that plasmid DNA, the same enzyme we'll use to cut these particular plasmids. Why same restriction enzymes? So that they can produce sticky ends, because if we use different restriction enzymes, so we cannot ligate these fragments with this plasmid, so recombinant DNA cannot be produced. So for that we have to use same restriction enzyme, because then only sticky ends can be produced and the restriction enzyme sites will be the same and DNA ligase can work. Now in these plasmids, these green colored plasmids, we have inserted these fragments. We'll insert these fragments. Now they become recombinant plasmids. Then these recombinant plasmids were transformed. They are transformed into E. coli cells. E. coli cells. So E. coli cells may, when we transfer these plasmids, so many E. coli cells were produced, and all the fragments of DNA, they are transformed in different E. coli cells, not only in a single E. coli cell. All these recombinant vectors which are having these fragments of cell genome DNA of which contain the fragment of DNA of these cells. Now they are transformed in a E. coli, and we'll construct the genomic library by using these E. coli cells. Of course, these E. coli cells contain recombinant DNA. Now there is recombinant DNA present in these cells, as well as its own genomic DNA, and many copies of these E. coli cells are produced, and we'll get multiple copies of these recombinant plasmids where whole DNA is inserted in different plasmids. This kind of library in which whole genomic DNA is used is known as genomic DNA library.